Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope you all will be fine and enjoying good health. I'm Dr. Muhammad Ali Hashmi from University of Education at a campus, and we are here to study the subject of Introduction to Biochemistry. This is lecture number 23, and in this lecture we'll study about significance of lipids in membrane transport mechanism. In the previous lectures we learned about significance of lipids in membranes, and then about fluid mosaic model of the membrane structure. So this lecture is just the continuation of those topics, and it will explain about which mechanisms are involved in the transport of materials across the membranes. The main concepts you will learn in this lecture would include the significance of lipids in membrane transport and different transport mechanisms, and then through those mechanisms involve three main types: passive transport, active transport, and then endocytosis and exocytosis. So in this lecture we'll be studying about the passive transport. So transport means anything that moves across the cell membrane. Either it moves from the extracellular environment to the intracellular environment, or cell discharges something from inside the cell to the outside world, and that includes the uptake of nutrients and the excretion of waste materials as well. So these all things come under the umbrella of membrane transport. So membrane transport is essential for cellular life, and as cells proceed through their life cycle, a vast amount of exchange is necessary to maintain the function. So the transport is always necessary to maintain the function of the lives. We need transport in our daily lives to transport and to send different things to different places, and then to keep the life functions running. Similarly, cell is a machinery which needs a lot of transport and exchange of goods to maintain its life functions. Transport may involve the incorporation of biological molecules and the discharge of waste products that are necessary for normal function. So as I told you earlier, transport can be incorporation of biological molecules, uptake of nutrients from the cell, and discharge of waste products that are necessary for the normal life and daily routine of the cell. Membrane transport refers to the movement of particles, that is solute, across or through a membrane barrier. So membrane transport can be linked to the term that it means the movement of particles, not only the liquid or the solvent form. It also moves particles or solute form across the membrane barrier. So membranes are normally selectively permeable. They are called as selectively permeable. What does that mean? Selectively permeable means it lets some substances pass through the membrane and stops others. So it is not like it's an open channel that everything can go inside the cell and can come out of the cell. No, it's a security controlled gate that some substances can pass through this and others cannot. So these membranous barriers in the case of the cell, for example, consist of a phospholipid bilayer. So what is that barrier? That is a phospholipid bilayer. It's a double layer of phospholipids. The phospholipids orient themselves in such a way so that the hydrophilic or polar heads are nearest the extracellular and intracellular mediums. So as we already studied about the fluid mosaic model, we know that the polar heads or they are the nearest the extracellular intracellular environment, and the hydrophobic or the non-polar tails, they align between the two hydrophilic head groups. So I'll just take you to the slide where the figure is present, and here you can see that the polar heads, they are either towards the cell or towards the outside wall. So this side shows the cellular environment, and that is the outside world. So you can see that these polar heads, which are depicted in these red balls, they are attached to the cell, and then they are attached to the outside wall. And the non-polar tails, these yellow long tails, they are between these polar heads. They align themselves between this phospholipid bilayer. Membrane transport is dependent upon the permeability of the membrane, transmembrane solute concentration, and the size and charge of the solute. So what are the main factors on which the membrane transport depend? That depends on these following three factors. So the first is permeability of the membrane, what is being allowed to go inside the cell and what is not allowed or what is forbidden. So it depends on that thing. The second thing is transmembrane solute concentration. If a solute has to move from outside the cell to the inside, what is the concentration of that solute inside the cell and outside the cell? So we have to look for that concentration. If the solute is moving against the concentration gradient, then definitely that process will require energy. But if it is moving along the concentration gradient, then that process will not require the aid of external energy. And the third thing is size and charge of the solute. If solute molecules are smaller, they will move easily and pass through the membrane. If their size is large, definitely that will pose a problem, and then there will be specific mechanisms to take those kind of solutes inside and outside of the cell. And similarly, their charge, if the charge on the particles can affect their entry or exit through the plasma membrane. So solute particles can traverse the membrane via three main mechanisms, that is passive transport, facilitated diffusion, and active transport. So some of these transport mechanisms require the input of energy and use of a transmembrane protein, whereas other mechanisms do not incorporate secondary molecules. So out of these mechanisms we'll study, some mechanisms will operate without the use of any energy of the cell, and some mechanisms will be there which require the use of energy because they're moving the particles or solute molecules against a concentration gradient. So if you move anything against a concentration gradient, you require energy for that. So here are the main mechanisms of transport. The first one is passive transport. Number two is active transport, then endocytosis and exocytosis. So in this lecture we'll cover the passive transport, and the rest of these three mechanisms will be covered in the next lecture. So we move to the passive transport. The most direct forms of membrane transport are passive. So it's common that many direct forms of the membrane transport, they work in a passive way. What is passive transport? It is a naturally occurring phenomenon and does not require the cell to exert any of its energy to accomplish the movement. So this passive transport is naturally occurring phenomenon which occurs naturally and commonly, and it do not use any of the energy of the cell, so it happens without the expense of any energy. In passive transport, substances move from an area of higher concentration to an area of lower concentration. So that is very easy and understandable. You should have studied basic science. So this is a fact that anything which has two different concentration gradients, it moves from the area of higher concentration gradient to the area of lower concentration gradient. Consider the example of air freshener which you just press in the corner of a room, and very soon it spreads to the whole room. So where you press the button of that air freshener bottle, in that area, when you release those molecules of the air freshener, its concentration was higher, but in the other parts of the room its concentration was 0. So it simply diffused through the air, and very soon you will see that all the molecules of that air freshener, they are in the whole room. So no energy was required, and that just spread throughout the room. So this way substances move from an area of higher concentration to the area of lower concentration. A physical space in which there is a range of concentrations of a single substance is said to have a concentration gradient. So wherever there are two different concentration gradients of concentrations of a same substance, like the air freshener in a place where it was sprayed and its concentration in the whole room. So whenever there are two different concentrations of a substance, that substance is said to have concentration gradient. So plasma membranes, as we already studied in the last lectures, that they are asymmetric. They are not symmetrical in structures. The interior of the membrane is not identical to the exterior of the membrane. So both sides are different in structures. And in fact, there is a considerable difference between the array of phospholipids and proteins between the two leaflets that form a membrane. So the inner leaflet, which is towards the cytoplasm, and the outer leaflet, which is towards the outside of the cell, they have different array of phospholipids, different arrangement of phospholipids and proteins between themselves. So the proteins and phospholipids, they are arranged differently around the two different sides of the plasma membrane. So on the interior of the membrane, some proteins serve to anchor the membrane to fibers of the cytoskeleton. So on the inside of the cell, some proteins are necessary which hold the membrane and bind it to the fibers of the cytoskeleton. If they don't hold the membrane can slip or move from there. So some proteins bind the membrane to the cytoskeleton so that it keeps in place and around the cell. So there are peripheral proteins on the exterior of the membrane that bind elements of the extracellular matrix. So you can see that the membrane is asymmetric because on the inside the structure is different and outside it is different. So there are proteins on the outside which bind the elements of extracellular matrix, which keep the cell in place when cells are joined together to form tissues. So these proteins bind the cell to the outside world. Carbohydrates are also attached to lipids or proteins, and they are also found on the exterior surface of the plasma membrane. These carbohydrate complexes help the cell bind substances that the cell needs in the extracellular fluid. So what is the function of those carbohydrates which we saw protruding out of the membrane? Just in that case, you can see that these little protrudings out of the cell membrane, What is their main function? They are there to bind some substances which are needed to be transported inside the cell. So these all things adds considerably to the selective nature of plasma membrane. So all these factors we just discussed, they add to the selectivity of the plasma membrane and make it selectively permeable. So now this figure has already been discussed many times in fluid mosaic model and just now. So for now we'll just have a look at it through the perspective of transport mechanism. So you can see that different molecules, like you see cholesterol, they are embedded in the plasma membrane, and then they are glycolipids with carbohydrates and glycoproteins with carbohydrates. So these beads are carbohydrates, and these bigger mass is protein underneath it. So these carbohydrates, they function to attach different substances which need to be moved inside the cell. Similarly, we have different protein channels which, like this one, you see these channels let different substances pass through the cell membrane. So we already discussed this point as well, that the plasma membranes are amphiphilic. I mean, they have both hydrophilic and hydrophobic regions. So as we just discussed, that these head groups, they are hydrophilic regions, while these tails are hydrophobic regions because they're non-polar. Heads are polar, they are hydrophilic. These tails are non-polar, they are hydrophobic regions. So this characteristic helps the movement of some materials through the membrane and hinders the movement of others. So this is the beauty of the cell membrane, that has both hydrophilic and hydrophobic ends, so it can control the materials which pass through it. Lipid soluble materials with a low molecular weight can easily slip through the hydrophobic lipid core of the membrane. So if something has a low molecular weight and it's soluble in lipids, that can easily pass through these barriers, these non-polar regions of the plasma membrane. But substances such as the fat-soluble vitamins A, D, E, and K, they readily pass through the plasma membranes and in the digestive tract and other tissues. So these vitamins, they are fat-soluble and they can easily pass through the plasma membrane in the digestive tract, like in the intestines, and where all the food particles get absorbed into the cells and into the blood. Fat-soluble drugs and hormones also gain easy entry into the cells and are readily transported into the body's tissues and organs. So the drugs and hormones which are fat-soluble, they get an easy entry into the cells. Molecules of oxygen and carbon dioxide have no charge and so pass through membranes by simple diffusion. So these molecules, small molecules which are uncharged, they can pass easily through the cell membrane by diffusion. Polar substances present problems for the membrane. So the substances which have polar groups, you just saw that we have these polar heads, and then in between them we have non-polar tails. So anything that needs to go through here, it needs to pass through these non-polar regions. So polar substances will have a problem here because they will not be soluble in fats or lipids, and they will face difficulty passing through the plasma membrane. So while some polar molecules connect easily with the outside of a cell, they cannot readily pass through the lipid core of the plasma membrane. That is what we just discussed. So additionally, while small ions could easily slip through the spaces in the mosaic of the membrane, so small ions have the capability, that they are small enough that they can slip through the spaces in that mosaic of the membrane, but the problem is that they have charges on them. So those charges prevent them from passing through the plasma membrane. So ions such as sodium, potassium, calcium, and chloride, they must have special means of penetrating plasma membrane. They should have some channels or gates through which they can pass. They cannot simply diffuse through the plasma membrane. So simple sugars and amino acids also need help with the transport across plasma membrane. Similarly, sugar molecules, they are also polar molecules and they need help to get transported across the membrane, and that is achieved by various transmembrane proteins or channels through which these sugar molecules can pass. Now we discuss about the diffusion. Diffusion is a passive process of transport. So a single substance tends to move from an area of higher concentration to an area of lower concentration until the concentration is equal across a space. So you see that this is a plasma membrane, and this in the bottom space is inside the cell. So if you look this substance, its concentration is very higher outside the cell, but its concentration is very low inside the cell. So it needs to pass through the membrane and balance its concentration across this membrane. So it just passes through its area of higher concentration to the area of lower concentration through simple diffusion, just like I gave you example of the air freshener diffusing into the room. So now you see that after simple diffusion, its concentration became equal inside and outside the cell. So that is how this process will take place. Here is a little video for you where you will learn about the diffusion process interactively.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
So materials, they can pass through the plasma membrane through the simple diffusion, and they move within cells cytosol by diffusion, and certain materials move through the plasma membrane by diffusion as well. Diffusion expends no energy. On the contrary, concentration gradients are a form of potential energy dissipated as the gradient is eliminated. So what are the concentration gradients? They are a form of potential energy which is dissipated or used when the gradient is eliminated, mean the concentration became equal on both sides of the membrane, then that potential energy is dissipated. Each separate substance in a medium, such as the extracellular fluid, has its own concentration gradient. So concentration gradient is not a universal phenomenon. It is different for each substance. So there are sugar molecules, there are other substances, there are solute particles, water, sodium, potassium ions. Everything has its own concentration gradient, and that is independent of the concentration gradients of other materials. So in addition, each substance will diffuse according to its own gradient. It will not have any effect or anything to do with the other substances and their concentration gradients. So within a system there will be different types, different rates of diffusion of the different substances in the medium. So each substances will be moving at its own speed depending on its own concentration gradient. Next is facilitated diffusion. In facilitated diffusion, materials diffuse across the plasma membrane with the help of membrane proteins. So facilitated diffusion helps those molecules which cannot simply pass through the plasma membrane due to certain reasons, like we studied for smaller ions which have charges on them. They are not allowed to pass through the plasma membrane. So a concentration gradient exists that would allow these materials to diffuse into the cell without expending cellular energy, but these materials are ions or polar molecules that are repelled by the hydrophobic parts of the cell membrane. So facilitated transport proteins shield these materials from the repulsive forces of the membrane, thus allowing them to diffuse into the cell. So those proteins, the facilitated transport proteins, just help these substances to pass through the membrane. So the material being transported is first attached to the protein or glycoprotein receptors on the exterior surface of the plasma membrane. So the first thing is the attachment of the material which needs to be transported inside the cell. So this allows the material that is needed by the cell to be removed from the extracellular fluid. So when the material is attached to those protein receptors, it is removed from the extracellular fluid, and then substances are passed to specific integral proteins that facilitate their passage. So first it is attached to the receptors outside the membrane, and then the substance is moved to the specific proteins which allow its passage through the membrane. So some of these integral proteins form a pore or a channel through the phospholipid bilayer. We just learned about these pores in here. You can see that they have a specific pore or a passage which let things pass through the bilayer. So there are some other carrier proteins which bind with the substance and aid its diffusion through the membrane. So some protein help move things through channels, and some proteins bind with the substances and aid in the diffusion process. So channels are specific for the substance that is being transported. This is important, that those channels are not universal, they're specific for the substances. Channel proteins have hydrophilic domains exposed to the intracellular and extracellular fluids. They additionally have a hydrophilic channel through their core that provides a hydrated opening through the membrane layers. So they have a hydrophilic channel on these sides which allow the opening to the intracellular and extracellular environment. So passage through the channel, these channels allow polar compounds to avoid the non-polar central layer of the plasma membrane that would otherwise slow or prevent their entry into the cell. So a polar molecule will definitely have problem here in the non-polar regions of the plasma membrane. So this protein is shielding those interactions, and a polar molecule will pass easily through this protein, which will stop its interactions with these non-polar parts of the phospholipid bilayer. So another type of protein embedded in the plasma membrane is a carrier protein. This aptly named protein binds a substance, and in doing so triggers a change of its own shape, moving the bound molecule from the outside of the cell to the its interior. So what this protein does, it binds the substance and then changes its shape and takes the substance into the cell and releases it there. So depending on the concentration gradient, the material may also move from inside the cell to the outside the cell. And carrier proteins are typically specific for a single substance. This selectivity adds to the overall selectivity of the plasma membrane. So as usual, these proteins are also selective and specific for different substances. So you can see that a substance binds here, the protein changes its shape and moves the substance here, and then it releases it inside the cell. So now for the facilitated diffusion, I have another animation for you. which will just clarify your concept about facilitated diffusion.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
So, if we summarize all these passive transport mechanisms we studied in this lecture, the first point would be that passive transport operates without the use of cell energy. Diffusion, what is diffusion? It is the movement of molecules from an area of higher concentration to an area of lower concentration. So that happens without the use of cell's energy. Facilitated diffusion, what is that? It is the movement of molecules through specific channels or carrier proteins and still without the use of energy. So let's conclude this by watching an animation which summarizes all these things together, and I hope that will increase the level of your understanding about this mechanism.
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins.
I hope this lecture and these animations would have increased your understanding of the passive transport and about how things and chemicals move across the cell membrane from inside the cell to the outside world and from the extracellular environment to the inside of the cell. So I have an assignment for you. It's a little question. It's a scenario you have to study, and then you have to select the correct option. This is about the permeability of the membrane. So it will show how much understanding you have gained in this lecture. So you can ask for the correct answer from your teacher in the Google Classroom. Here are the references that were consulted for the preparation of this lecture, and I wish you the best of luck for your studies. We'll see you in the next lecture with more transport mechanisms, and until then, Allah Hafiz.